Alright, so let's go back to this. We use transcription by using RNA polymerase to make a RNA copy of DNA. When it's finally done, it's going to attach itself. So when you realize you look at the actual strand of messenger RNA before it leaves the nucleus, it has a lot of parts that we consider to be literally called junk DNA, or that are not necessary. Well, we have the ability to actually, or our cells have the ability of removing the unwanted parts. Sort of like splicing commercials out of a movie. The parts that are unwanted are called introns. The parts that you do want, that are called active regions of a gene, are called exons. And so we have enzymes that splice the introns out and reattach the edits of the exons. When I'm all done, I have a fully functional, no commercials added version of messenger RNA. Now I have the exact code to make that protein. But, well, in certain extent, they are nucleotides, but we have learned that they simply don't play a role in the overall protein structure as it exists today. So some people hypothesize that they are either older versions of that gene that evolution said this is no longer functional in this time frame. For example, if we became a lot colder, maybe we need to increase our metabolic rate at a faster pace. Maybe that idea to make your cells work faster and make more heat was found in this part of the DNA. But now that we live in a warmer environment, evolution said, you know what, if we could simply not give us this extra boost of energy requirement, we can run at a cooler temperature and be more comfortable in a cooler environment. And so that's what we think introns can be, is either A, unnecessary STRs, or versions of the gene that are parts of the gene that were needed a long time ago, or you can argue that it's evolution's way of saying, you know what, you may need this possibility up in the future. And so rather than trying to make a mutation happen, this random pattern allows us to maybe be able to do something we want to be able to do in the future. That makes sense? It's a great question. And part of my answer is we don't fully understand that, but we have some guesses as that when you compare this to some other primitive animal, it has a little bit of its intron in our exon, and we realize we don't need that ability, so it's evolution saying we can get rid of it. Yes, maybe to be a lot more functional, because then we're going to have hiccups in the structure of our amino acid sequence, which may make a little more sense when we get here in a second. All right, so now our messenger RNA, right, is going to leave the nucleus. What are these holes called? Nuclear pores, right. So come on in and have a seat right there. I won't bite it. Come on.
Sorry.
He's hiding, trying to record it. That's all right, fine.
How does it know that it's not moving?
Well, so we're leaving an awful lot of this process out. It's a really complicated thing. There are enzymes called operons that allow for the operation of actual production of fully formed messenger RNA. And these operons, for lack of a better way, know that there's a certain sequence of nitrogenous bases that cause it to let go, to detach from the DNA. And when the operon detaches, then the RNA polymerase right behind it will also then detach. Like it cuts, yeah. So, let's get to here.
Sorry, I had a quick question about the cap and tail. Why is that added?
I have no idea why it's added. All right. Again, it's one of those things where we don't understand why the cells do what it does, but we know that to a certain degree it allows for a certain stability to it, because what's called the poly A tail, which goes on the 3' end, you guys don't really need to know this. It can be as many as 300 adenines bound in a row. Just A. Everyone, what the hell are you doing this for? It's like a busted record. But it does seem to make the overall thing a little more stable than when you find mutated messenger RNAs that don't have that many A's on the end of it. How does it know to do that? Nobody knows. It just happened to have a bunch of A's on the end of it, and you survived better than her version who only had half the number of A's. And that's what evolution is if your version of something is better at something than hers will die out, and yours will increase in number. And now all we see is the current version. We have no idea how many attempts nature went through to make messenger RNA stable. Some could have had 1,000. And went, well, that's a bit overkill, that's too many. So only had 20. We need more than that. And it's ended up being a number that we found to be about 250. All right? So we leave the messenger, we leave the nucleus. We then go to the rough ER, because remember, that's where we make proteins. But most importantly, it's the ribosomes that are on the rough endoplasmic that makes the protein. Now we have two places to find ribosomes. They can be fixed to the ER, or they can be free-floating all by themselves. So they're floating ribosomes. And the ribosome is the factory for making the protein. Let's see how we do this. All right, so transcription is where we make a strand of messenger RNA from a strand of DNA using RNA polymerase. That leaves the nucleus after being modified and edited. Now I'm going to go to a ribosome. Well, a ribosome is really a big hunk of RNA. It's also an enzyme. Now, here is my strand of messenger RNA. Each one of these little tabs represents a nitrogenous base, right? An A, a C, a G, or a U perhaps. If you look at the cartoon, pay attention to the detail. There's these three prongs and a bit of a space. That space is bigger than that space. So if you look at the pattern, threes, threes, threes. We call the groups of threes a codon. So it is a collection of three nitrogenous bases. And it's the code for that amino acid. The codon tells the ribosome which amino acid will be put here. Now, the ribosome is a big RNA structure. How do I get the amino acids to the ribosome in the first place? That's where what's called the tRNA comes in. These guys. And the T stands for transfer. Because they're going to transfer the amino acid from the cytoplasm of the nucleus and bring it to the ribosome. So the T makes sense. We're transferring amino acids to the growing protein strand. We have codons, which are the regions of messenger RNA that are a pattern of three nucleotides that will code for that amino acid. So here's the codon for aniline. Well, here comes the newly arriving glycine, and that's the codon that codes for glycine. What I'm thinking about is that these are a sequence of nitrogenous bases, right? So we're going to go from a sequence of nitrogenous bases, A's, U's, G's, and C's, to a sequence now of amino acids. So we're translating from a nucleotide language to an amino acid language, if you like. And it's the ribosomes and transfer RNAs that allow us to do that.
So, where are the tRNAs carrying the amino acids?
Well, these guys are all out in the cytoplasm.
Where you said that they're carrying the amino acids
From the cytoplasm.
Okay.
Yeah, so in a sense, let's do this. Each one of you is going to be a ribosome, and you're on ER. So this is rough ER. Let's put helium balloons, thousands of them, in this room floating around. And we're going to give each of you a letter of the alphabet, all right, or whatever. We'll make you an amino acid, all right? So we're going to have alanine, glycine, and tyrosine in the back. So we've got three separate codons on my messenger RNA. So group of three nucleotides, group of three nucleotides, three nucleotides. I'm a transfer RNA. I will randomly bump into the amino acid that I am designed to carry. It's random. So I bump into my amino acid. Now I'm floating like a helium balloon in this room. All right? Let's say I'm going to be for alanine. But I bump on the other codon that's over here. Well, if this is an enzyme, think what enzymes do, right? They help chemical bonds to form. We need to make a peptide bond between this amino acid and this one. If I land over here, first of all, I'm not going to be able to make a bond anyways. I'm going to detach. But if I also land over here, and this is for a different amino acid than I am, I won't attach. Because this part right here called the anticodon is the complement for the codon. So if this was an AAA, this would be a UUU. That's how this transfer knows to attach there. And it has to be in the ribosome, not a UUU over here, or I'm sorry, an AAA. When we look at the amino acids that are here, this is what a tRNA actually looks like. Do not memorize this. This is just to make maybe a little more sense of the structure. Now we can actually see that it is a strand of messenger RNA, or of RNA, because there's a U in there first of all, so I know it's RNA. And because of its hydrogen bonds, it forms what we call a clover leaf pattern. Well, here's the anticodon down here. It is the one part of the transfer RNA that has the opposite complement to the codon on the messenger RNA. Up here is where the amino acid attaches. It doesn't attach anywhere else. So what you have is basically a taxicab service or a shuttle service. It's transferring amino acids to the place where you actually make the covalent bond happen to form the peptide bond. That would be the actual ribosome itself. To understand all of what evolution has done in terms of how many transfers we have, let's skip that one for a second. I think I want to try something a bit different. Now, this is what's known as the codon chart for amino acids. Do not memorize this. You're going to use it, yes. And if you go into high-level biochem, you will memorize this. But what it lets me realize is, if I have a codon of UUU, then that's the code for phenylalanine. Then you realize, all right, there's only really two different codons that would code for phenylalanine. Come down over here, I've got 2 codons for leucine. Oh, I've got 6 codons for leucine. There is a specific transfer RNA for each one of these, and only for one. So I'm the transfer RNA for UUG. I will only attach to